what is it made up of, and what are its functions? What is a cell membrane?
All the stuff in
Yep, seals in all the juices. Good. All right, what's it made up of? Good. Phospholipids. Good. So there's a lot of fat, right? Fat molecules. Make up the membrane. Good. And what does it do? What are some of the things it does? It does a lot of things. Name some of them.
it. Keeps the cell from falling in, like maintains the strength of the cell.
Okay, good. The integrity of the cell, good. Keeps it all together. What else?
Selectively permeable, allows some things in, some things out.
Good. It's kind of a gateway, right? You have to go through it. Some things can't. And good. It has a lot of other things too. So it has these cell recognition markers, right? To help differentiate different cells and identify that it's not a foreign cell. Good. Also, it's got a bunch of channels and pumps to help regulate electrolyte differences inside and outside the cells. Good. Those are some things. Good. Understand the different organelles, where they are, and what they contain and do. What are some of the organelles?
Ribosome.
Mitochondria, ribosome, lysosome. What is it?
Lysosome.
Yep, lysosome, Golgi, nucleus, endoplasmic reticulum. Good. All right, let's start at the top. Ribosomes. What do they do?
Protein synthesis.
Yeah, they're like protein making machines. That's what they do. Good. Yep. Made up of protein and RNA. And they can be either in one of two places. Yeah. Exactly. They can be floating around freely, you know, not committed to any one place. Or they can be bound on the rough endoplasmic reticulum. That's the RER. Okay? Good. And again, they provide enzyme linked activity to make proteins. Now let's talk the endoplasmic reticulum. You got two types. The rough and the smooth. The rough makes proteins, right? Because what makes it rough? Yeah, all the ribosomes that are stuck to it makes it look grainy and sandpapery, rough. So that makes proteins that you're going to excrete. So this would be like hormones, antibodies, different things that are going to get secreted out into the blood are made in the rough endoplasmic reticulum. Okay? The smooth ER makes lipids. Good. Very good. And it doesn't have any ribosomes on it, therefore it appears more smooth. Good, good. All right, moving on. All right, the Golgi is known as the post office of the cell, right? It receives packages, puts their little markers on it, and sends them where they need to go. Okay? And how does it send things away? Yeah, in a vesicle, which are basically a little membrane pod that holds whatever is being sent, and it goes and delivers it where it needs to go. Good. All right. Organelles, Golgi apparatus, and vesicles. Alright, let's skip that slide. Mitochondria, the powerhouse of the cell. It makes protein, ATP. Yeah. Good. And we're going to talk, did we talk about this? length?
Cellular
Like the
respiration.
glycolysis?
Yeah.
Yes.
We did, right? Okay, yeah. Happens in the mitochondria. Good. Lysosomes and peroxisomes are similar but different in what they do exactly. So they're both kind of the cleanup crew. Think of them like that. Lysosomes, lysol. That's what I remember. So they digest proteins, carbohydrates, nucleic acids, a whole bunch of stuff. Garbage disposals of the cell. Peroxisomes do a similar thing, only they digest lipids, alcohol, and hydrogen peroxide. Okay? Good. La, 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 la. All right. Good. And then the centrosome. Let's just talk about that real quick. What's the centrosome good for?
Producing spindle fibers?
Yeah.
Things that split
The split. Yep. The thing is to split the things. Exactly. So this is really they're only active during cell division, right? So whether this is meiosis or mitosis, and we'll talk about that later. But centrosomes, they just make the spindle fibers that help to split the chromosomes. All right. Good. All right. Next. Know the bolded terms and definitions given throughout this lecture, so don't forget that. And understand the structure of DNA, how it is stored in the nucleus, and the concept of gene expression. Okay, so DNA. Let me tell you about its structure.
Double strand.
Yep, double-stranded in a helix shape,
Composed of
like a twisted bladder. Sorry, what did you say?
I was trying to think about the sugar.
Sugar? Okay. Yeah, so it's made up of nucleotides. All right, and the nucleotides have phosphate, sugar, and a base. Right? Good. We'll get there.
DNA really isn't discussed a whole lot in chapter 3 or chapter 4.
Oh, we gotta misplace that. Alright. Alright, well let's go. Chapter 4.
Are we, the chapter 3, are we gonna need to know more about the cell cycle though?
Yeah, so know the phases. Know generally what's going on in different phases and what happens during those phases. And then kind of what happens during interphase between, which is basically cell gets ready for the next round, synthesizes its DNA during that phase. Okay?